All right, so now that you guys have a good idea of what the amino acids are and their primary and secondary structure, let's talk more about the genetic code and how many nucleotides are actually necessary to specify or code for an amino acid. So, a couple of important things right off the bat. First of all, the genetic code is in triplicate form or triplicate code. So, three nucleotides are going to code for each amino acid. And so we call this a codon. So, because that genetic code is in triplicate code, there's three nucleotides that are going to come together and designate each amino acid. Of the nucleotides that are present in messenger RNA, those are going to be A, G, C, and U, or adenine, guanine, cytosine, and uracil. So there's four nucleotides that are possible to make up this codon or this genetic code. So within each of those different codons, codon 1, 2, and 3, there's four different possibilities. Therefore, we get 64 different codons. And that's really what we see in this figure here. So again, same idea as the figure in the previous section, but here we can actually see more of our coding and start and stop proteins. So methionine is always going to be our start protein and is encoded for by AUG. And then our stop proteins are one of three, and they don't have a specific name other than just a stop protein. So they are amino acids, but they don't have like a specific name like phenylalanine or leucine. So codons are going to be set up in a couple of different ways. So keep in mind the codon is going to be three nucleotides in a sequence together, and of those 64 possible codons, one is going to be the start codon or methionine, and then there's going to be three of those stop codons that we just saw on the previous slide. Therefore, there is going to be a total of 61 sense codons that code for amino acids. So this really leads to the concept of a genetic or a degenerate code, and that's a term that was actually borrowed from the physics discipline. And what it means is that there are multiple physical states that can have an equivalent meaning. So that's why we see codons that are going to code for the same amino acid or synonymous codons. So for example, threonine here in the center, two of the nucleotides are going to be the same, the first base and the second base. So if you're dealing with threonine at all, the first two codons are always going to be AC. We still get threonine, and it doesn't matter if that third position is a U, a C, an A, or a G. So this is what we mean by a degenerate code, is that we can have multiple states or multiple codons actually coding for the same amino acid. So amino acids are going to be basically brought in to start forming the polypeptide chain by transport RNA. So each transfer RNA is going to bind one amino acid. Okay, so as we get moving here, you're going to see a lot of figures. So tRNA refers to that transfer RNA, and amino acid is going to be designated by amino acid or the actual 3-digit code that is shown in this figure here. So transfer RNAs are going to possess an anticodon, and that anticodon is going to match the codon on the mRNA. So when you hear the word anticodon, just think of the opposite codon. So transfer ribonucleic acids are going to be material that is made up of ribonucleic acid material. So A, G, C's, and U's, they're going to be bound up and twisted around to form these kind of funky transporters. And so that means that they are not protein, but they're still material that has been produced from DNA. So their very specific purpose is that they are going to be that transport RNA that is going to serve as the mechanism to bring in amino acids and form polypeptide chains on the depending on what the sequence is of the mRNA strand. So just to set up this figure for you, down here at the bottom, this green line here is oriented 5' to 3'. Thus, that is the messenger RNA that was produced from transcription. And within that messenger RNA is going to be nucleotide codes. Okay, so A, G, C, and U. So for example, the code on the messenger RNA for this first position is UCC. That means that the transfer RNA that has the anticodon or the opposite code of AGG is going to is going to identify the messenger RNA, come in, set down right on that codon, and is going to bring in the amino acid that it actually codes for. So each transfer RNA codes for a very specific amino acid. So just really quickly, we can also see that down here towards the right side of this figure, we've got our messenger RNA, and the second code within that messenger RNA strand is UCU. So a different code than the first one, but it's actually a degenerate code because U and C are going to, in the first and second position respectively, are always going to code for serine. So we can get any of these combinations of U, C, and one other nucleotide in the third position, and they're all going to code for serine. Therefore, this transfer RNA, because it has a very specific anticodon for either AGG or AGU, that is going to be very specific to serine. So whenever we see that type of coding happening in the messenger RNA, it's always going to match up with serine. So this third position here is what we call a wobble position. And this actually creates a wobble effect because this transfer RNA has the ability to recognize either a G or a C or an A or a U. So it doesn't matter. It's because it's going to be matching up with serine. It's going to be bringing serine in. So it really doesn't matter what this third position is as long as it's one of these four total codes. So this wobble position actually allows some transfer RNAs to pair with more than one codon on a messenger RNA strand. So we see here that in our first position, we've got C, G, A, and U. And then in our third position, if we're going to have a G, then it's going to pair very specifically with one of these two anticodons and codons. If we've got a C or a U, that's where we get the wobble effect going. So there's between 30 and 50 transfer RNAs that exist in our cells, and they can actually pair with the 61 sense codons. So we don't actually have 61 different sense transfer RNAs. There's only 30 to 50 of them because more than one of them can actually bind to multiple triplicate code on the mRNA strand. All right, so let's take this concept just a little bit further and talk about reading frame and initiation codons. So a reading frame is going to be the RNA or the messenger RNA sequence. So if this is our nucleotide sequence up here at the top, AUA, GCA, GUC, that is actually going to be split up into 3 different reading frames or 3 different codons. So codon 1, codon 2, codon 3. We can get some overlapping to take place. So this is where we start to see position shifts. So if codon 1 is misread, it could actually, codon 2 could actually be UAC rather than CGA if we get base pairs that are changing. And that's going to become really important when it comes to phenotypic expression. So keep in mind that AUG is the initiation codon. Every single starting factor of translation is going to start with AUG, methionine, and then that's going to start the polypeptide chain. So our termination codons are going to be one of 3, UAA, UAG, and UGA, and these don't code for an amino acid. They actually are not going to have a transfer RNA that's actually has an anti-coding sequence for these 3 termination codes. So if the polymerase is or the transfer RNA is actually going along and reads one of those, it's not going to synthesize a RNA or an amino acid. It's just going to stop with the transcription process or translation process. All right, so we'll pick up here in the next segment.